formally called the second lecture, is to talk about integrating these ideas into one example. And so the one example is going to be detecting a biological lipid, which is a very important biological lipid called endotoxin. So before I do that, are there any additional questions? Yeah.
So in the part 2, I will talk about the phase separation. Does that only happen when you don't have sufficient surfactant in the system? If you like overload it with all the places?
Yeah. So if you saturate the surface, then that's right. You'll drive the interface into a homogeneous state. But one can, for example, look at lipid mixtures. And so you may have a density of lipid at the interface that is close to saturation coverage, but now the phase separation is the segregation of the components of that lipid monomer. And in fact, the general rule of thumb is that biological membranes operate on the edge of miscibility gaps. And that the reason many of the functions of biological membranes come about is because you segregate and you create inhomogeneous membranes. And so in order to be close, in order to achieve that sort of structuring of the membrane, you have to be very close to miscibility gaps. And so that's true in this system also. If you have a mixed monolith on the surface, you can be close to phase separation, and the interaction of the lipid crystal can put you over the edge of that past that miscibility gap.
So the optimized theory you mentioned in the liquid crystal and liquid crystal interface is the same as the old one?
Well, and in fact, there is no Hofmeister series, but there are a family of Hofmeister series, plural. And so, depending on what biophysical phenomena we're looking at, there are different rankings of ions. And so there's a general overall trend that you'll tend to find these highly polarizable anions like chlorate and cyanate at one end, and then you'll find these small, highly hydrated ions at the other end. But the actual ranking changes depending upon the phenomenon that you're looking at. And so in broad strokes, yes, we see a very similar sort of ranking that you would see, for example, in protein solubility, salting and salty active proteins. The exact ordering of some of these anions changes depending on the phenomenon. And what's going on here is that these so-called chaotropic ions, they actually absorb and penetrate into the interface of the liquid crystal. And so they perturb the organization of the liquid crystal through that penetration at the interface. Any other questions? So what time should I be leaving?
Well, we should delay the end of the 15 minutes and perhaps 10:45.
Okay. So this example application of liquid crystals involves the oil-water interface, but not the planar interface that I was talking about earlier, but curved interfaces associated with droplets of liquid crystal. And so we're talking about emulsions, and so these are oil and water emulsions, where the oil is the liquid crystal. And as I indicated here, you have ionic phenomena occurring at these interfaces, and so the things which I've just discussed earlier are going to manifest themselves in this system. So if you have droplets of liquid crystal in water, the liquid crystal within the droplets can take on a variety of different configurations, and these have been well studied in literature. If you have tangential anchoring of the liquid crystal, you'll tend to form this so-called bipolar configuration, and so you have two surface defects, woojums, at each of the poles of alternatively, if you have a homeotropic anchoring of the liquid crystal, the droplet will, or can at least, take on this radial configuration where you just have now a point defect at the center of the droplet. And so if you add an adsorbate, such as a surfactant, that changes the anchoring of the liquid crystal, you can navigate your way between these two extremes, and that's what's shown on this slide. And so the adsorbate is sodium didecyl sulfate, so it's a surfactant, very similar, opposite charge to the cationic surfactant I was describing earlier. And so as you increase the concentration of the surfactant in aqueous solution, as shown down here, in the millimolar or 10th millimolar range, you see this transition between the two limits, the bipolar and radial configuration, through these intermediate states, which have associated with them an internal disclination loop and this so-called pre-radial configuration with a point defect associated with the surface. And so these are actually equilibrium states of the system, and as you add and subtract surfactant, it adjusts reversibly to the surface of the droplets, and you can navigate your way along this path. So this is SDS, and so nobody really cares about a sensor for sodium didecyl sulfate. But I want to tell you about an amplifier people do care a lot about, and that's so-called endotoxin. So this is an illustration of the outer surface of a bacterium. It's a so-called gram-negative bacterium. It has two cell membranes, and the outer membrane is shown here, and it has associated with it these so-called lipopolysaccharides. So lipo means lipid, and so there's a lipid component that keeps it anchored into the membrane, and then it has a big polysaccharide head group. And so the lipid component is called lipid A, and that's what's shown down here. And so unlike the phospholipid I was talking about earlier that has two tails, this one has 6 tails. And so it's a very interesting lipid, so a unique structure. And our immune system is tuned to detect it. Why? Because if you have a bacterial infection, the bacteria will shed lipopolysaccharide, which is also called endotoxin, and your immune system will pick up that endotoxin and tell you that you have a bacterial infection and mount a defense against it. And so it's a very important lipid. For the same reason, you don't want to have endotoxin associated with, for example, a drug that you're taking, a intravenous injection, or a biomedical device, because if you do, and you have an intravenous injection and it's contaminated with endotoxin, then your body will mount an immune defense against that injection, and you'll end up with a fever. In the worst case, you end up with, in this is an extreme case, sepsis. And so that's if you have a bacterial infection that's generating a lot of endotoxin, and that can lead to death. And so that's a very extreme case. But because of the immune response to endotoxin, in the United States, the agency that regulates drugs, the FDA, requires that all intravenous fluids, all biomedical devices have to be assayed for endotoxin. And you have to establish that endotoxin is below about 25 picograms per ml. So that's the sort of the relevant concentration. And if you just look down here, these are all the different contexts in which people are measuring the presence of endotoxin, and these are the concentrations that you have to detect it at. And so the one that I'm talking about here is sterile water for injection or irrigation, so 25 picograms per ml. And so how's it done at the moment? And so how it's done at the moment is shown here. And some of you might recognize that if you're from the east coast of the US. And so that's the horseshoe crab. And so the horseshoe crab is very clever. And so it's evolved a defense against infection. And the way it defends itself against infection is to coagulate its blood. And that's because then you isolate the region of the infected body, because you don't have the blood circulating through it. And so back in the 1980s, some scientists on Cape Cod worked out that you could capture horseshoe crabs and bleed them, and then you produce the blood, or a lysate prepared from the blood, as a reagent in a test for endotoxin. And so it works actually very nicely. So you take your little sample of blood, you pop your water for injection into it, and if the blood coagulates, then you've got endotoxin in your sample, so you don't have to worry. And so that is now the gold standard for detecting endotoxin. And what happens is that there's a collection of enzymes in the blood, and so when you add endotoxin, you trigger this cascade of enzymatic reactions that results in clotting, ultimately. And so it's very nice, but if you're using enzymes, of course, you're susceptible to interference of the enzymatic activity. So, for example, these enzymes rely on bivalent cations. So if you have chelating compounds in your solution that you're interested in assaying, they will interfere with the enzymatic activity. It's a biologically derived reagent, and so as the seasons vary, the blood of the horseshoe crab varies. And so there's a lot of calibration that has to go into performing assays. And of course, the horseshoe crab is limited in supply. But the good news is that after they're bled, they're put back in the water to live a happy life. So anyway, so this is a very nice assay, and it was a big advance on what existed before the assay, and that was the rabbit. And so before the horseshoe crab assay, you injected a rabbit with your sample, and you waited to see if the rabbit developed a fever. And if the rabbit developed a fever, then you have endotoxin in your sample. And so we and others are very much interested in seeing if we can find substitutes, other ways of detecting endotoxin. And given that endotoxin is comprised of lipid A, and so what you really want to detect is the lipid A, we thought, well, maybe we could use anchoring transitions involving liquid crystals to detect the presence of this particular liquid. So we started looking at the appearance of these droplets of liquid crystal in solutions of endotoxin. And indeed, one can drive a droplet from a bipolar configuration in the absence of lipid A or endotoxin into a radial configuration in the presence of endotoxin. And so this experiment being done here is an experiment at a microgram per mil of endotoxin. And just remember that the relevant concentration is 25 picograms per mil. And so this is nice, but it's not the sensitivity that you require. And so we then started diluting the endotoxin, and we found something that was very surprising. And so this is reporting the fraction of droplets in the sample that exhibit the radial configuration as a function of the concentration of endotoxin in solution. And so what we found was that you can go down into the picogram per mil concentration regime and actually trigger these transitions in configuration of the liquid crystalline droplet. And in fact, this is too sensitive, and so you can detune the system by just changing the density of liquid crystalline droplets in your dispersion. And so you can design a system that responds to the tens of picogram per mil concentration range, which is what you want. So it looks sort of promising, but then if you do a simple calculation, you see something which is actually very, very surprising. And so let's do a calculation of how many endotoxin molecules are involved in these configurational transitions of the droplets. And so in a typical experiment, we're working with about 0.16 nanoliters of 5Cb that we disperse just by sonication into 1,400 droplets. And then we're dealing with 40 microliters of a picogram per mil endotoxin solution, which corresponds to about 481,000 molecules. And so take the number of endotoxin molecules and divide it by the number of liquid crystalline droplets, and you end up concluding something very strange, and that's that we're actually triggering these configurational transitions with, on average, something like 300 endotoxin molecules per droplet. And so how many endotoxin molecules does it take to saturate the surface of one of these droplets? It takes of order 10 to the 7 molecules. And so what we discovered was that you can drive these configurational transitions through interaction with endotoxin at surface coverages of these droplet interfaces, which are about 10 to the minus 5 below saturation monolayer coverage. And this is actually a concentration of endotoxin on the surface. We really expect to impact the anchoring energy. And so we went into this project thinking that we were going to be looking for a response based on an anchoring transition, but what this suggests is that that's actually not the case. And so indeed, if you do another experiment where you create a planar interface of the liquid crystal. Yeah.
So in your previous, so did you also observe this intermediate state, like a disclination loop?
So the answer, that's a very good question. So the answer is no.
Okay.
And
So
actually,
only hedgeable observed.
what's that?
So you only observe the hedgeable observed.
Yeah, in fact, so like, to cut to the end story here and tell you that it's not a surface-driven anchoring transition, but it's a transition occurring through another mechanism. And consistent with that, if you look at the transition states, which we have done, you don't see the same kinetic pathway when you trigger a configurational transition with endotoxin as you see when, for example, you use SDS. And so two different mechanisms and actually two different pathways. So how much endotoxin do you need on the surface in order to change the anchoring of the liquid crystal? And so we determine that by transferring endotoxin onto a planar interface, and I'll spare you the details in my time. But what we did was we spread a monolayer on the surface of water, we transferred it onto the liquid crystal, and what we determined was that you need about 1 lipid A molecule per 1.3 nanometers squared in order to change the anchoring of the liquid crystal. And so this is quite a high surface coverage. It's close to saturation surface coverage, and we're many orders of magnitude below that level of coverage. And so this is also evidence that we're in fact not looking at a surface-driven anchoring transition in this case. And then the other very interesting observation we made was the selectivity to this particular lipid structure. And so what I'm showing here is a plot of the concentration of the 4 lipids or surfactants shown on this slide required to trigger the configurational transition. And so for lipid A, we're down in the picogram per mol range. But for these other lipids, so these double-tailed lipids shown here and here, and for sodium dodecyl sulfate, you're up in the 10 microgram per mol range. So about 6 orders of magnitude higher. And if you do a little calculation, at this concentration, you have sufficient surfactant or lipid in solution to saturate the surface and change the configuration through the surface anchoring transition. And so this requires about 10 to the 7 molecules per liquid crystalline droplet. This configurational transition is occurring at order 1,000 molecules, several hundred molecules per droplet. So the question is why? And so we've been looking at confinement of liquid crystals in droplets. And so let me tell you a little bit about how confinement influences the configuration of a liquid crystal. And so the message here is going to be that the elasticity of the liquid crystal sensitizes the system to the adsorbent. And so if you have a droplet of liquid crystal dispersed in an aqueous solution, you can ask, how does size affect the configuration of the liquid crystal in the droplet? And the configuration is dictated by this competition between surface anchoring and elastic energies. So the elastic energy scales as the elastic modulus times the radius of the droplet. The surface contribution scales with the surface area, so the square of the radius. And so you see this crossover when the size of the droplet is of order of micrometer. So for large droplets, the term that goes as R squared, surface anchoring dominates. You expect the bipolar configuration. For small droplets, the term which goes linearly in size, you expect to dominate. And so you see minimization of the elastic energy, and so you see this homogeneous configuration. And so this is what you would expect to happen on the basis of this scaling argument as you decrease the size of the droplet. So we set out to test that prediction, and so what we did was we made droplets of different size, and the details I don't have time to describe to you, but in brief, what we did was we fabricated polymeric capsules and we filled them with liquid crystal. And so we could keep the surface chemistry constant, and we could vary the size of the droplet by changing the size of the capsule, and the capsule was synthesized using a silica particle template. And so it allowed us to make droplets, and they're actually encapsulated droplets in this case, of very well-defined size, and this was done with a collaborator in Australia. And what you see here are optical micrographs of droplets 10 micrometers in size, 85 down to about 700 nanometers in diameter. And so we asked, what's the configuration of the droplet inside? And what we saw was not what is predicted by that very simple scaling argument, but what we saw instead was that as you decrease the size of the droplet, you go from this bipolar configuration into a pre-radial and ultimately into a radial configuration. And so this was a bit of a surprise to us, and it caused us to go back and think, well, what could be driving this radial configuration as you decrease the droplet size? And what we have focused our attention on is the role of saddle splay elasticity. And one can write down the free energy of the bipolar and radial configurations, including K24, this saddle splay elastic constant. And if you look at the contribution that it makes to the free energy of the radial and the bipolar configuration, what you see is that it stabilizes the radial configuration. And so if you look at this free energy for going from the bipolar to the radial configuration, you have K24, which for a sufficiently large value of K24, and it needs to be larger than 1.5 K11, can stabilize the radial configuration. And what we're saying here is that this change from the bipolar to the radial configuration is not coming about because of a change in surface anchoring. So the easy axis is still tangential to the surface, so this favors the bipolar configuration. But take K24, according to the scaling argument, can push the system into the radial configuration against that surface anchoring energy. And so if K24 is larger than 1.5 K11, then at some critical radius, which is given by the difference between these two quantities over the anchoring energy, you would expect to have the radial configuration being the lowest free energy configuration. And so this is a proposition, and an open question is, what is the magnitude of K24? And so there's an Erickson limit on its magnitude, and it's actually 1.53 K11. So there would be a very narrow range of K24 values that would allow you to predict the stabilization of the radial configuration, but it's also just an order of magnitude argument. And so what we're suggesting is that in these droplet systems, K24 is predisposing the system to switch into the radial configuration. And that this is why the system responds to low concentrations of lipids, much lower concentrations than you can drive anchoring transitions at planar interfaces. And so if it's true, you would expect the response of these droplets to endotoxin to be strongly dependent on the size of the lipid crystalline droplet. And indeed, we do see that to be the case. And so this is the fraction of droplets in the radial configuration in response to 100 picograms per ml of endotoxin. And once the droplets get to a diameter that's greater than about 10 micrometers, the sensitivity of the system to endotoxin is lost. And so these elastic energies, this is our hypothesis, is that they're winding up these droplets in a manner that makes them predisposed to undergo the transition to the radial configuration. And indeed, from this simple sort of energy balance, one can make an estimate of the influence of the endotoxin on the free energies of these droplets. So once the droplets get to a size which is about 10 micrometers in diameter, the imbalance of elastic and surface anchoring energies is sufficient that the binding of endotoxin cannot overcome the stabilization of the bipolar configuration. And so if one plops in a radius of 5 micrometers into this free energy balance, you end up concluding that the influence of the endotoxin on the free energy of these droplets is about 10 to the minus 17 joules. So what does that mean? So if you look at the critical aggregation concentration of endotoxin, and so this is the concentration of endotoxin in solution at which you'll see the endotoxin molecules self-associated, so it's the equivalent of a critical micelle concentration. From knowledge of that critical concentration, you can calculate a standard free energy of self-association of endotoxin. So it's telling you about the driving force for endotoxin undergoing self-assembly. And so if you take the critical aggregation concentration, which is around 10 micrograms per mil for endotoxin, and plop it into this expression, what you end up concluding is that if 700 endotoxin molecules self-associate, you can generate a free energy of around 10 to the minus 17 joules. And that's sufficient that we calculate to trigger that configurational transition. And so this number is right in the ballpark of what we estimate to be the sensitivity of this system. And so what we believe is going on in this system is that the self-association of endotoxin with these liquid crystalline droplets is driving this configurational transition. So it's not just absorption at the surface, but it's a type of self-assembly that's going on. And so in light of time, I think I'll just finish up by doing one last thing. And so this is sort of our free energy balance. And so what we're saying is that if these elastic and surface anchoring terms are very close to being in balance, then the standard free energy of association of endotoxin is sufficient to drive the configuration transition. So size puts the system out of balance, but also changes in anchoring energy will put the system out of balance. And so this comes back to the idea of ions. And so if we change the ion composition of the system, we expect to change the anchoring energy. And indeed, we see the response to endotoxin change with ionic strength of the aqueous solution in a manner that's consistent with the contribution of the electrical double lag to the anchoring energy, and also with the pH of the system. And so sort of this returns to the idea that one can manipulate the anchoring energy and the response of these liquid crystalline systems to biological adsorbents through changing the ion composition of the system. And so I'll skip the last few slides and just go to the conclusion and say And we went into it actually thinking we were going to be looking for a surface anchoring transition. That turns out not to be the case, but there's another type of association going on between the liquid crystalline droplets and the lipids. And we have some evidence that what's actually going on is that the lipid is localizing at the defects of these droplets. And if you look at the radial configuration after exposure to endotoxin, you find evidence of the endotoxin at the center of these droplets. And so we've done that by fluorescent labeling. And so these are highly sensitive and specific things, and it's an assay that we're busy at the moment translating into a commercial product. So there are a number of unresolved issues, and so let me just end with this. And so, we've invoked the idea of K24 in stabilizing these droplets. The value of K24 for 5CB is not unambiguously determined, and some estimates of K24 suggest that it's insufficient, in fact, in our system to stabilize the radial configuration. Another question is, to what extent do these double layer contributions to anchoring energies depend on the size of the droplets? And so the device screening lengths are hundreds of nanometers in size, and so there may well be a size-dependent contribution to the anchoring energy that's coming about from these ionic phenomena. And then something that I just alluded to a minute ago is that there's a process of self-assembly of lipids occurring in the regions of defects in these systems. And something that we're currently working on is to understand how the structure of the amplifier really dictates the self-assembly of the amplifier in the regions of these defects. And then the students in my group who've been involved in this work with endotoxin are underlined in this slide, and we have a very nice group of collaborators. And one of the things that you quickly learn if you want to research that sort of straddles physical and biological sciences is that you need good biologists as collaborators, and we have some terrific people from the life sciences who have enabled this work, and this is the sponsorship. And with that I'll stop and be happy to take any additional questions if there are questions.
Thank you for this great interdisciplinary talk. I think you've heard this question loads of times already. But if you have, for example, yogurt and you want to measure the, for example, the endotoxin, and you want to apply this to this method, what is the influence of other compounds besides endotoxin on, for example, these observations?
Yeah, so
that sounds difficult. Because a lot of other compounds present in your material.
Yeah, and so so that's a very good question. And we know that there are some compounds that interfere with these assays. And one of the things which, so maybe there's two parts to my answer. So the first is that what we're looking at the moment is detecting endotoxin in relatively simple things, like intravenous fluid. So all you have to do there is salts. Well, it's mainly salts. And actually, what we have been able to show is that if you take these droplets dispersed in PDS, phosphate buffer, you can take a sample, and if you dilute it 100-fold into PDS, then the ionic composition of your sample gets diluted, and it has no influence on the response. So the fact that we have such a high sensitivity allows us to dilute samples, and so that means that we can dilute potentially interfering compounds. And so that's okay for salts, but things like yogurt, where you have a very high concentration of protein, that may be a challenge. And so we're not looking at things that complicated. And serum would be another one. So serum, you would like to be able to detect endotoxin from serum, but that's a very complicated protein-rich mixture. And so whether one can navigate the interference of the protein content of serum in a liquid crystalline system that's up in the air. So, and you know, I can tell you also that even for the crab blood-based technique, things like serum create a problem. Is the lipopolysaccharides bind to the proteins also, and so the response of this crab blood-based assay is influenced by the binding of the LPS to serum proteins.
Short technical question. What challenge would be having none of the production of nanoscale size droplets? For example, a few hundred nanometers per micron. Which method do you use to produce this droplet size and control the size?
Yeah, so you're right that so what we're doing, so we looked at microfluidic-based methods to produce droplets in order to study the size-dependent configurations. And our experience is that to go below about 5 micrometers is actually very hard with microfluidic methods. And so what we resorted to was what's shown here. And so you start off with a silica template particle, and those you can buy with very tight size distributions. Actually, quite small. I mean, we didn't do a lot below about 700 nanometers, but other people have with this approach. And so you take the template particle and you wrap it in a thin polymeric film, which is prepared by sequential deposition of polyelectrolytes. You then etch out the silica core, and then you fill it with liquid crystal. And this is actually a process you do in a beaker. And so we can prepare 700 nanometer capsules filled with liquid crystal. We stop there because we can't characterize what's going on optically inside them. But you can fabricate, and there are examples in the literature of these capsules being fabricated down into the 100 nanometer size range. And so you're really governed by the size of the template particle. So it's actually a very nice way of making monodisperse droplets. And we also made encapsulated droplets of isotropic oils. So it's not
Use it by yourself.
using the same technique that was published on it.
Okay, so we'll have to stop for coffee break. Coffee is outside, and we'll come back at 10 past 11. So let's thank the speakers before we go for coffee.